Okay, so welcome back to this next video in which we are discussing the vascular endothelial growth factor receptor. Okay, so we've just discussed the PI3 kinase AKT-mTOR pathway, which is one of these pathways that is activated by these phosphotyrosine residues on the cytoplasmic side of the vascular endothelial growth factor receptor 2. Okay, now what we're going to discuss is another pathway which is going to be activated by these phosphotyrosine residues. Now, basically, it starts again with another enzyme coming and binding to the phosphotyrosine residues. And in order to do this, this enzyme has to have an SH2 domain. So here we have an SH2 domain, another SH2 domain, which will come along and bind to another phosphotyrosine residue. Okay, so you're also going to recruit this enzyme to the cytoplasmic domains of your vascular endothelial growth factor receptor 2s. And this enzyme is by the name of phospholipase C gamma. Okay, so it's a phospholipase C type enzyme, and specifically it's a phospholipase C of the type gamma. Okay, now this is not a normal type of phospholipase C. If you know phospholipase Cs, the type you'll be thinking of is the one that's downstream of the G protein, and basically that's a phospholipase C of the beta type. So the type that's going to come and bind to these phosphotyrosine residues with an SH2 domain is a phospholipase C of the gamma type. However, the enzyme pretty much does the same thing as the beta type, okay, because they're both phospholipase C enzymes. And basically, again, this enzyme is now positioned just underneath the lipid bilayer, basically, right next to its target within the cell membrane. And its target is actually exactly the same target as phosphatidylinosyl-3 kinase. Phosphatidylinosyl-3 kinase added a phosphate group onto the third carbon of the inositol ring of PIP2 molecules. Phospholipase C gamma is going to break down PIP2 molecules. So let's draw out another PIP2 molecule here. So let this be the lipid bilayer here, and here is a PIP2 molecule. So here are the two long-chain carboxylic acid molecules esterified to the glycerol molecule, which then has a phosphate group coming off it with the inositol ring, and this time only two phosphate groups. Remember, we're talking about PIP2, phosphatidylinosyl 4,5-bisphosphate. Okay, so let's colour these in. The long-chain carboxylic acids will be coloured in orange. The glycerol molecule is here in green. Okay, the phosphate groups, all three of them, will be coloured in vivid purple. And then the inositol ring will be coloured in blue. Okay, right. So, basically, the phospholipase C gamma enzyme is going to work on these molecules within the phospholipid bilayer, and it's going to cut the bond between this phosphate group and the third alcohol group of the glycerol molecule. So remember, the phosphate group is linked to the alcohol group of the glycerol molecule via a phosphoester link. You're going to cleave that, you're going to hydrolyze it, and you're going to get two products. One of them will remain in the phospholipid bilayer. This one where you have a glycerol molecule which now has only two of its alcohol groups occupied by these two carboxylic acid molecules. This will remain in the phospholipid bilayer, okay, because it's still anchored there by these two long-chain carboxylic acids. Okay, and this molecule is what's known as a diacyl glycerol molecule. Okay, and for short, diacyl glycerol is often abbreviated to DAG. Now, there is another name for diacyl glycerol molecules. They're also occasionally known as diglyceride molecules. Okay, so either term is acceptable. Right, so the diglyceride or the diacyl glycerol molecule will remain in the lipid bilayer. However, this other molecule that you create, which is the inositol molecule with the three phosphate groups bound to it, this has no reason to remain by the phospholipid bilayer. It will wander into the cytoplasm, basically. and this molecule is called inositol, and then we've got 3 phosphate groups coming off it. And remember, you would not name this 1, 3, 4, because remember, we're talking about a specific optical isomer of inositol. We're talking about myoinositol, and the carbons of myoinositol are already labelled up for you. So this is 1 here, 2, 3, 4, 5. Okay, so this is inositol, okay, 1,4,5 trisphosphate. And if you were being really proper, which hardly any people ever are, you would call this myoinositol 1,4,5 trisphosphate. So if you ever do see that, if you ever do see it called myoinositol 1,4,5 bisphosphate, sorry, not bisphosphate, trisphosphate. Sorry, that's a terrible mistake. You've got 3 phosphate groups coming off there, inositol 1,4,5 trisphosphate. If you ever do see it referred to as myoinositol 1,4,5 trisphosphate, don't be afraid of it. It just refers to the optical isomerism of the inositol ring, and myoinositol is the normal optical isomer of inositol that is used in cells. Okay, and inositol 1,4,5 trisphosphate molecules are usually just abbreviated to IP3. The I is for inositol, the P for phosphate, and then because we've got 3 of them, it's IP3. Okay, right. So both of these molecules are going to have downstream effects, basically. The IP3 is going to diffuse into the cytoplasm, and it's going to act on receptors for IP3 molecules, which are in the membrane of an intracellular organelle. So let's draw this intracellular organelle here. Okay, so this intracellular organelle, which contains receptors for inositol 1,4,5 trisphosphate, is called the endoplasmic reticulum. Okay, and the endoplasmic reticulum is usually just abbreviated to the ER. Okay, and the endoplasmic reticulum, amongst other very important roles within the cell, is a store of calcium, basically. You have a lot of calcium ions stored within the lumen of the endoplasmic reticulum, and you have very, very low levels of calcium in the cytoplasm of the cell. In fact, calcium level within the cytoplasm of the cell is around 100 nanomolar, which is tiny. If you compare that to the concentration of potassium ions in the cytoplasm, which is 150 millimolar, okay, so millimolar is 1,000,000 times bigger than nanomolar. So this is around 1.5 million times bigger, basically. Calcium concentration is minute within the cytoplasm, basically, is the message from that. Okay, so you do, however, have a much, much higher concentration of calcium in the ER lumen. And basically, if you open up little channels across the ER membrane, then calcium can leave the ER lumen and go into the cytoplasm. And this calcium will then act as an intracellular signal and will cause all sorts of downstream effects. And basically, this is what these IP3 receptors in the surface of the ER are. They are calcium channels. Now, usually they are closed, but when IP3 binds to them, this can lead to them opening. Now, it doesn't lead to them directly opening, but indirectly, the inositol 1,4,5 trisphosphate does lead to them opening. So we're not going to go into the workings of the IP3 receptor in detail here, but IP3, it's an reasonable simplification to say that IP3 will lead to the opening of the IP3 receptors. In reality, it doesn't do it directly. If you bind IP3 to the receptor on its own, that will not cause it to open. But it does, it's a useful simplification to assume that it leads to the opening, and it is true, basically. It does lead to the opening, just not directly. Okay, so here are the inositol 1,4,5 trisphosphate receptors. Okay, so when IP3 goes up in the cytoplasm of the cell, it will cause these IP3 receptors indirectly to open, and you will then get calcium released from the ER membrane into the from the ER lumen into the cytoplasm of the cell. Now, this calcium and this diacylglycerol, those will be capable of activating a certain enzyme which is at the phospholipid bilayer, basically. So I'm going to draw this enzyme here. Okay, so it's attached to the inner leaflet of the phospholipid bilayer. Okay, so it's got lipid molecules which anchor it into the phospholipid bilayer. And this enzyme is known as protein kinase C. So we're very lucky today we get to talk about not only protein kinase B, but also protein kinase C over here. So, for short, protein kinase C is usually abbreviated to PKC. Now, protein kinase C not only needs the diacylglycerol to activate it, but it also needs the calcium ions that are being released from the ER to activate it. So together, the diacylglycerol and the calcium activate protein kinase C, and once you've activated protein kinase C, then it is a serine threonine kinase enzyme. Now, what is the target for protein kinase C? Well, basically, it's going to phosphorylate an enzyme known as sphingosine kinase. Okay, so let's talk about this down here. So, we're going to phosphorylate a molecule. Whoops, what am I doing? I've forgotten the S, never mind. Cancel that out. Okay, so sphingosine kinase is what's going to be phosphorylated. And specifically, it's sphingosine kinase 1. And for short, sphingosine kinase 1 is often abbreviated to SPK1. Now, some people will abbreviate that. Will put an extra letter in here. Some people, instead of putting SPK1, will put SPH for sphingosine kinase 1. So SPHK1. Either of those are permissible, basically. Now, what does this enzyme do? Well, basically, it converts another normal component of the lipid bilayer, known as sphingosine, into sphingosine monophosphate. Okay, so let me show you the structure of sphingosine. Right, so let's start with the structure of sphingosine then. Right, so sphingosine's proper name is, it's called 2-amino 4-octadecene. Okay, octadecene. And then we also have 1,3-diol. So this is the proper name for sphingosine, 2-amino 4-octadecene 1,3-diol. Okay, so this tells us that we've got an 18-carbon hydrocarbon where you have one double bond here, and that's between the 4th and the 5th carbon. You also have an amino group off the 2nd carbon and alcohol groups coming off both the 1st and the 3rd carbon. Okay, so I think we can now draw that molecule. Now, the only important carbons here are the 1st, the 2nd, the 3rd, the 4th, and the 5th. We don't care about the other 13. Okay, so I'm not going to draw out the whole 18 carbons. I'm only going to draw the first 5 and then abbreviate the rest of them. Okay, so let's draw out the first 5. 1, 2, 3, 4, 5. And then the rest of it, what will the rest of it be? Well, what you're going to have is 12 methylene groups. So we've got 13 carbons left. 12 of them are just going to be in methylene groups. So because I don't want to have to draw 12 methylene groups out, I'll draw one methylene group and then put a bracket around it and a suffix 12. That's a useful trick to get out of having to draw loads of methylene groups. And then on the end you have to put the methyl group separately because it's not a methylene group. So that then takes care of those other 13 carbons that are boring, basically. And now, between the 4th and the 5th carbon, we have a double bond, so let's put that in. Of these carbons, you'll then have one hydrogen each, so we'll put those on. Okay, and then off the 3rd carbon, we know we have an alcohol group, so we might as well put that in. And we also will have a hydrogen. Off the 2nd carbon, we'll have an amino group, we might as well put that in, like so, and also a hydrogen. And now, off the 1st carbon, we have an alcohol group. So I'll put this in here, with 2 hydrogens also. So, this is the structure of sphingosine, or 2-amino 4-octadecene 1,3-diol. Okay, now as you can see, this is a very long molecule and it's quite hydrophobic, at least this bit down here is. So what can happen, well, what does happen is this implants into the phospholipid bilayer basically. So you have this molecule within the phospholipid bilayer. Now the enzyme sphingosine kinase, which usually is not active, but once the protein kinase C enzyme has been activated, protein kinase C will stick a phosphate group onto the side of sphingosine 1 kinase. So, if we just try and connect everything up with arrows, the protein kinase C will now stick a phosphate group onto this sphingosine kinase enzyme, and I will denote sphingosine kinase just as SPK, and this is sphingosine kinase 1, so SPK1. Okay, so let's colour in that phosphate group in purple, like all the other phosphate groups. And this sphingosine kinase 1 enzyme is now going to convert sphingosine into sphingosine 1 phosphate. Okay, and to do that, it's going to add a phosphate group onto the alcohol group that comes off the first carbon. So it's going to stick a phosphate group on here. Okay, and this phosphate group will be attached just like all the other phosphate groups that we have seen that have been attached onto alcohol groups. Okay, now then follows something that is not understood basically. What happens is somehow the activation of sphingosine kinase 1 triggers the activation of something known as the RAS G protein. Okay, so I'll put this here. So there is a very famous monomeric G protein. So we've seen other monomeric G proteins. We've seen REB, for instance. Now REB isn't that famous, not compared to RAS. RAS is an extremely famous monomeric G protein, and there isn't just one of them, there are actually 4 of them, but we won't get bogged down with that. We'll just abbreviate them all as RAS. Okay, so RAS is a monomeric G protein. So as I described previously, it has 2 states. It can either be in the off state, where it has guanosine diphosphate, GDP bound to it, or it can be in the on state, where it has guanosine triphosphate, GTP bound to it. Okay, so that's the case for all G proteins, that you have 2 states, an on state and an off state. Okay, now, we do not know how, but the activation of sphingosine kinase 1 triggers the movement within the cell towards an on state for RAS, basically. So you start getting more RAS, which is in the on state rather than in the off state. So somehow it leads to this, and we think that it leads to it not by activating the conversion of the off state into the on state, but by inhibiting the conversion of the on state into the off state. So we think somehow that the activation of sphingosine kinase 1 leads to the inactivation of the reverse process of the inactivation of the RAS G protein. Okay, but we haven't elucidated this pathway yet, and whether it involves the phosphorylation of the sphingosine, again, we don't know basically. Probably is the answer. Okay, so sphingosine kinase 1 then blocks the activation of the sphingosine kinase 1 converts the sphingosine into sphingosine 1 phosphate, and sphingosine 1 phosphate, we think, stops the somehow leads to the blocking of the conversion of RAS GTP into RAS GDP. So the levels of the on RAS G protein go up within the cytoplasm of the cell. Okay, and the RAS G protein binds to an enzyme known as CRAF, also called RAF1. Okay, so I'll put this here. So here is the enzyme CRAF. Okay, and CRAF also has another name. It's also known as RAF1. So basically, this enzyme is usually inactive, and it's a serine threonine kinase once it is activated, and it's activated by the binding of the RAS G protein, which occurs when the RAS G protein is in the on state. So here is the RAS G protein with GTP bound to it, which we know has now gone up in the cell, and it's going to bind to the CRAF enzyme. Now, to make this simpler, I'm going to colour in CRAF in orange here. Okay, now what does CRAF in turn do? Well, CRAF, as I said, is a serine threonine kinase, so it's going to phosphorylate serine and threonine residues, and one of its targets is an enzyme known as MEK, okay? And it's going to stick 2 phosphate groups onto MEK, and they're both put onto serine residues. So you add on 2 phosphate groups onto the MEK enzyme, and both of these are onto serine residues, and it is the CRAF or RAF1 enzyme which does this. Okay, so this enzyme is known as MEK. Okay, now MEK is short for MAP kinase, or in fact, actually, I'll explain this in a moment. We'll just say MAPK, okay, /ERK, ERK. So this is what the M is for, MAPK. You take the M, and that's the M here. Then the E is for ERK, and then the final K is for kinase. So this is MAPK, ERK, kinase. Okay, so that's MEK. Now, MEK itself is a kinase enzyme, but it would not be true to say that it is a serine threonine kinase, and also it would not be true to say that it's a tyrosine kinase either. Basically, it's going to add phosphate groups onto both a tyrosine residue on the next enzyme along, and also onto a threonine residue, so it's neither one nor the other, it's both. Okay, so the next enzyme along then is MAP kinase or ERK. Okay, so it's now going to add 2 phosphate groups onto the next enzyme along, and this is why this is called a kinase cascade, because basically we've had kinase phosphorylate another kinase, which then phosphorylates a third kinase. And one of these phosphate groups will be onto a threonine residue, and the other will be onto a tyrosine residue. Okay, so what's this final enzyme called? Okay, well, this final enzyme is called MAP kinase or ERK. Okay, so we'll discuss what those 2 names actually stand for. So, this you can either call it MAPK, which stands for mitogen activated protein kinase, or you can call it ERK, which stands for extracellular signal regulated kinase. Okay, so let's go over the page and talk about these silly names. Right, so MAPK stands for the mitogen, that's the M, A is for activated, and then the P is for protein, and then the K is for kinase. Okay, so a mitogen is any any signaling molecule which induces mitosis, basically, induces cell division. So this is a protein kinase, so it's a kinase enzyme, and it is in fact a serine threonine kinase that is activated by mitogens, so it's a name that makes sense. Now, often this is just abbreviated to MAP kinase. People will often just call this enzyme MAP kinase, so they won't usually abbreviate the K. When they're writing it down, they will abbreviate the K. They will write MAPK, but people don't generally say MAPK. When they're speaking about this enzyme, they'll say MAP kinase. Okay, right. Now, annoyingly, this enzyme has another name, basically. It's also known as ERK, and ERK stands for extracellular signal regulated kinase. So the E is for extracellular, and then the S doesn't get anything, so signal doesn't get a letter, and then the R is for regulated, okay, and then finally the K is for kinase again. So this is the extracellular signal regulated kinase. So when people refer to this enzyme, often they will say MAP kinase ERK, okay, and they will call this entire pathway involving MAP kinase ERK, they'll call it the MAP kinase ERK pathway. Now, we aren't really seeing a conventional example of the MAP kinase ERK pathway because we've seen it activated by sphingosine 1-phosphate, which is not a conventional way for it to be activated. Usually it's activated by RAS guanine nucleotide exchange factors, which rather than inhibiting the inactivation of the RAS G protein, they activate the activation of the RAS G protein. Okay, so we're not really seeing a conventional example of it, but this is the MAP kinase ERK pathway. At least we're seeing the downstream portion of it. Right, okay, so we've activated this serine threonine kinase, which is known as MAP kinase or ERK. Okay, and we now want to see what is this going to do. Well, basically this enzyme goes into the nucleus, okay, and it phosphorylates transcription factors. Now, which two transcription factors does it phosphorylate? Well, there are two very, very powerful transcription factors which it phosphorylates. It phosphorylates MYC transcription factors. So here is MYC, and it's stuck a phosphate group onto it. MYC is a famous transcription factor. It's often called the cell's most powerful mitogen. Okay, and then another very famous transcription factor, less famous than MYC maybe, but still famous, is ELK1. Okay, so you phosphorylate these two transcription factors, MYC and ELK1. Now, we'll come back to MYC in a moment. We'll discuss ELK1 first. Okay, so ELK1 is a transcription factor. We've already discussed transcription factors. We know that they increase the expression of certain genes and decrease the expression of other genes. Now, one of the genes that ELK1 is going to increase the expression of is called CFOS. Okay, so you're going to get more CFOS. Now, CFOS then associates with another protein that will be in the nucleus, known as C-jun, to create CFOS-C-jun heterodimers. And this transcription factor basically does very similar things to MYC, and they again do very similar things to the mammalian target of rapamycin complex 1. So again, we'll come back to what MYC does and what CFOS-C-jun does right at the end, where we discuss what the mammalian target of rapamycin complex 1 does. So we're collecting our list of transcription factors, and I've just reminded myself of that song, Collecting Your Jar of Hearts, that one. So we're collecting a list of transcription factors, and there's one more that I want us to get before we then discuss what these transcription factors are going to do. So bag all of these transcription factors, bag MYC, bag CFOS-C-jun, and we've already got in our bag the mammalian target of rapamycin complex 1. And you might also like to include ELK1 also in a sort of half-hearted way, because ELK1, yes, it does lead to the upregulation of CFOS, which then leads to another very powerful transcription factor being produced, these CFOS-C-jun heterodimers. But that's not all it does. It's got a good 100 genes, which it increases affects the expression of. I shouldn't strictly say increases, because it could decrease the expression of some. But it affects far more than just CFOS-C-jun, so it's going to play a role as well. Okay, so we're now going to look at one last pathway, and thankfully it's much shorter than the others that we've seen. Okay, so basically we now go back to this step here. We've released calcium from the ER. That calcium is not only just going to activate the protein kinase C with the diacylglycerol and cause the pathway that we've just discussed, but it's also going to activate another pathway. Okay, and it's going to do this by activating a protein known as Calmodulin. Okay, so let's have a little discussion of the structure then of Calmodulin, because most people have heard of Calmodulin, but if you're asked to, make a comment on the structure of it, they wouldn't have a clue, and it's a shame because it's a very simple protein. Okay, so basically, Calmodulin consists of two lobes. Okay, with a nice little linker between the two lobes, like so. Okay, and these two lobes are known as the C lobe and the N lobe. Okay, and both the C lobe and the N lobe have two binding sites for calcium. So a calcium ion combined here, a calcium ion combined here, and this N lobe also has two calcium binding sites. Okay, so in total there are 4 calcium binding sites, and therefore in total 4 calciums combined. Now at the moment there's no calcium bound in any of these calcium binding sites. So the molecule is in this sort of hunched over shape. So when there's no calcium bound to the calmodulin, the whole molecule is in this sort of hunched over shape. I've sort of tried to draw it with this hunched back, basically. And the two lobes are sort of folded back towards one another. This structure is actually called apocalmodulin. So when calmodulin has no calcium bound to it, it's called apocalmodulin. And the shorthand for apocalmodulin is to call it apo, capital C, lowercase a, uppercase m. So capital C, lowercase a, uppercase m is the shorthand for calmodulin. Okay, now what can happen is 4 calcium ions can come along, bind to these 4 calcium binding sites on the calmodulin protein, and this changes the structure of the calmodulin protein. The whole thing springs out, basically. The two lobes move further away from one another, and the linker between the two lobes goes from being a linear polypeptide to being a alpha helical polypeptide, like so. Okay, so let me now show 4 calcium ions bound in the calcium binding sites, and I'll show these just as green dots. So 1, 2, 3, 4. Okay, so this structure then now is known as a calcium calmodulin complex. Okay, and for short, calcium calmodulin complexes are abbreviated to Ca2 plus CAM. So calcium is abbreviated to Ca2+, and then of course we use our abbreviation for calmodulin here, like so. So this is a calcium calmodulin complex. So we're going to get the production of these calcium calmodulin complexes when the calcium is released from the ER, and these calcium calmodulin complexes are now going to activate certain enzymes, and one of the enzymes that they're going to activate will show over here. Okay, so we've got our calcium calmodulin complex here, like so, and it is bound to a certain enzyme here, and this enzyme is known as calcineurin. Okay, so here it is, bound to calcineurin, and I'll just colour it in. I'll put those calcium ions in because they're the important parts. So we have a calcium calmodulin complex bound to this enzyme, and this enzyme is called calcineurin. Okay, and for short, calcineurin is often abbreviated to capital C, lowercase a, capital N, like that, so CAN. Okay, now calcineurin is a serine-threonine phosphatase. It removes phosphate groups from serine and threonine residues. Now, so it's the opposite basically of a serine-threonine kinase. It is going to work on a transcription factor known as NFAT. Okay, and NFAT stands for the nuclear factor, so that's the NF. N is for nuclear, F is for factor, and then the AT is for of, and then the A is for activated, and then the T is for T cells. So this is the nuclear factor of activated T cells. Now, the nuclear factor of activated T cells has a special region, which I'll show here, which is what's known as the serine-rich region. Okay, and usually in a cell, you do not have this nuclear factor of activated T cells active. And by the way, this is another transcription factor. So the role of this is to go into the nucleus and, affect the transcription of a whole bunch of genes by binding to promoter regions. However, usually in cells where the nuclear factor of activated T cells is not active, it cannot get into the nucleus. It is trapped in the cytoplasm. And the reason it is trapped in the cytoplasm is that it has a bunch of phosphate groups attached to serine residues within the serine-rich region. Okay, so these trap the nuclear factor of activated T cells in the cytoplasm, and now hopefully you can see what's going to happen. We have just activated calcineurin over here, which is a serine threonine phosphatase. It cuts phosphate groups off serine and threonine residues. So it's going to come over here, cut these phosphate groups off those serine residues, and therefore activate the nuclear factor of activated T cells, because suddenly it will lose these phosphate groups, and now it will be able to move from the cytoplasm into the nucleus, and it will act as a transcription factor. So now we've got our final transcription factor, the nuclear factor of activated T cells. So we've now collected up 4 or 5 transcription factors. We've got the nuclear factor of activated T cells, we've got C-phos-C-Jun heterodimers, we've got MYC, we've got the mammalian target of rapamycin complex 1, and we've also got ELK1 up here. Okay, in the next video we'll discuss what these 5 or 4 transcription factors are going to do.